right, so active transport. So, in what cases will we see active transport occurring instead of passive transport? So, in this picture here, you can see that passive transport, like we previously discussed, is when solutes go from an area of high concentration to an area of low concentration. So, you can see this through simple diffusion or facilitated diffusion, and for water, you see that through osmosis. Active transport, we're moving solutes from an area of low solute concentration to an area of high solute concentration. So keep in mind that doesn't flow as easily. That requires energy in the form of ATP from the cell. So, in what cases would we use active transport requiring energy from the cell when some substances must pass through the membrane? So, maybe those substances are too large, or those substances are moving from an area of low solute concentration to high solute concentration. We would need to use active transport in order to take in that solute and take it into the cell. So once again, active transport, we have solutes going from an area of low concentration to an area of high concentration. So we call that going against the concentration gradient. So because it goes against the concentration gradient, this does require energy from the cell. So we're going to talk about three different types of active transport. The first type is going to be used for small molecules. Oh, before we do that, here's just a quick summary of active transport. We have solutes going from an area of low solute concentration to an area of high solute concentration. And because we're going from low to high through this protein pump, we need ATP in order for this to work. So all types of active transport does require energy expenditure from the cell itself. All right, so molecular transport. This is transferring small molecules and also small charged particles called ions across the membrane. So because they're very small, they can fit into these protein pumps. This specific protein pump is a proton pump, but normally this is a protein pump and it's activated by ATP. So ATP helps activate the protein pump, and then solutes can go from an area of low solute concentration to an area of high solute concentration. So we're seeing that solutes are going against their concentration gradient. So in this picture, we have low solute concentration on the left side, high solute concentration on the right side. So for molecular transport, we see solutes going from an area of low to high with the help of protein pumps and also ATP in order to power the protein pumps. All right. The second type of active transport is when we're trying to take in larger molecules into the cell. So this is called endocytosis. So the first thing that happens is the membrane will wrap itself around the substance that it is interested in taking into the cell. After it engulfs the substance, it's eventually going to bud off into a vesicle. This is now inside the cell and it's also enclosed in a membrane, so it's not just floating everywhere. So, two different types of endocytosis. The first one is phagocytosis. So, this is anytime you're taking in large food molecules. So, you're engulfing large food molecules. An example of this is when an amoeba, this is an organism, consumes a bacteria. So, when the amoeba consumes the bacteria, it's consuming it through the process of endocytosis. It's wrapping its membrane around the substance of interest, which is the bacteria. Endocytosis, there's also pinocytosis, which is going to be the same thing, but in liquid. So when liquids need to be absorbed by the cell, or at least go through the cell membrane, but it's very large. Pinocytosis is a process where these liquid substances can be engulfed and enter the cell that way. So keep in mind, a lot of this with phagocytosis and pinocytosis, you can see that the cell is using energy in order to wrap itself around the substance that it is interested in taking in. So although it's not as extravagant as just ATP powering a pump. Here we can see that the cell is using energy in order to wrap itself around the substance. Last one is exocytosis. So this is opposite from endocytosis. So this is how we take substances out of the cell. So if it can't simply go across the membrane, it can't go from low to high concentration. We need to just get it out. The first thing that happens is the vesicle. So the vesicle is a membrane-bound organelle that surrounds the substance, is going to fuse with the phospholipid bilayer of the cell membrane. So it's going to attach to the phospholipid bilayer of the cell membrane. And then here you can see that once it fuses together, the membrane's going to come back together, and then it can use its energy from the cell to push out the substances. So it's almost opposite of endocytosis. Here you can see that it's wrapping around the substance and taking it in. For exocytosis, we are fusing with the membrane and then kicking out the substances.